Scientist Toolbox. This is a part of a series of international webinar series conducted by the Postgraduate and Research Department of Maharajas College, Ernakulam. This talk aims at providing key inputs into the research work on blood cancer going on worldwide. I am confident that you will find it an interesting program and give more insight into the research areas of blood cancer. I wish you a happy learning from this seminar. And now I welcome Dr. Jayamol Kivi, our beloved principal, for the inaugural address. Thank you.
Good morning, everybody. കേൾക്കാൻ പറ്റുന്നുണ്ടോ നിങ്ങൾക്ക്?
കേൾക്കുന്നുണ്ട്.
ഞാൻ കോളേജിലെ ഉള്ളിലാണ് ഇരിക്കുന്നത്, അതുകൊണ്ട് അതിന് റേഞ്ചിന്റെ എന്തെങ്കിലും പ്രശ്നമുണ്ടോ എന്ന് അറിയില്ല. സുവോളജി, മഹാരാജാസ് കോളേജിന്റെ സുവോളജി ഡിപ്പാർട്ട്മെന്റ് ആരംഭിക്കുന്ന ഒരു വെബിനാർ സീരീസിന്റെ ആദ്യത്തെ സെഷനാണ് ഇന്ന് നമ്മളിവിടെ തുടങ്ങാൻ പോകുന്നത്. തീർച്ചയായിട്ടും സമകാലിക പ്രാധാന്യമുള്ള വിഷയങ്ങളെ കുറിച്ചും എല്ലാവർക്കും വളരെയധികം ഉത്കണ്ഠയും ആകുലതയുമുള്ള വിഷയങ്ങളാണ് റെയർ ഡിസീസസ്. അപ്പം അത് വളരെ നേരത്തെ തന്നെ, നമുക്കറിയാം, വളരെ നേരത്തെ തന്നെ ഡിറ്റക്ട് ചെയ്യപ്പെടുകയാണെങ്കിൽ ചികിത്സ കൊണ്ടുമൊക്കെ ഫലപ്രദമായി മാറാവുന്ന പല വിധത്തിലുള്ള ഡിസീസസ് ആണ് നമ്മളിപ്പം ഈ ക്യാൻസർ പോലെയുള്ളത് എന്നൊക്കെ നമ്മൾ വായിച്ചിട്ടുണ്ട്. എനിക്കതിനെ കുറിച്ച് സുവോളജിയുമായി ബന്ധപ്പെട്ട വളരെയധികം നല്ല കാര്യങ്ങളൊന്നും അറിയാത്തത് കൊണ്ട് തന്നെ വിഷയത്തിലേക്കൊന്നും കടക്കാനുള്ള ഒരു പ്രാവീണ്യമൊന്നും എനിക്കില്ല. നമ്മുടെ സുവോളജി ഡിപ്പാർട്ട്മെന്റിന്റെ രമ ടീച്ചറും ഷർമിള ടീച്ചറും ഒക്കെ എൻ്റെ അടുത്ത് പറഞ്ഞു, ഇന്ന് നമ്മുടെ ഒരു വെബിനാർ തുടങ്ങുകയാണ്, ടീച്ചർ ഉണ്ടാവണം എന്ന് പറയുന്നത് ഞാൻ വളരെ സന്തോഷത്തോടെ ഇപ്പോ നിങ്ങളുടെ കൂടെ ഉണ്ടായിരിക്കുകയാണ്. ഏറ്റവും ഫലപ്രദമായി വിഷയം കൈകാര്യം ചെയ്യാൻ പറ്റുന്ന ആളുകളെ ആയിരിക്കും നിങ്ങൾ സെലക്ട് ചെയ്തിട്ടുള്ളതെന്നും എനിക്ക് മനസ്സിലാവുന്നുണ്ട്. തീർച്ചയായും ഈ വെബിനാർ ഇതിലെ എല്ലാ പാർട്ടിസിപ്പൻസിനും, ഞാനിപ്പോ നോക്കി ഒരു 150-150ഓളം പാർട്ടിസിപ്പൻസ് ഉണ്ട്. ഈ പാർട്ടിസിപ്പൻസിനൊക്കെ ഏറ്റവും ഫലപ്രദമായ വിധത്തിൽ അത് പ്രസന്റ് ചെയ്യപ്പെടാനും അവരിലേക്ക് കാര്യങ്ങൾ എത്താനും കഴിയട്ടെ എന്ന് ആശംസിച്ചുകൊണ്ട് ഈ വെബിനാർ സീരീസിന്റെ ഉദ്ഘാടനം നിർവഹിച്ചതായി പ്രഖ്യാപിച്ചുകൊണ്ട് എല്ലാവിധ ആശംസകളും നേർന്നുകൊണ്ട് നിർത്തുന്നു. താങ്ക് യു ഷർമിള. രമ ടീച്ചർ, താങ്ക് യു.
Good morning, one and all. A very warm welcome to the international webinar on understanding genetic predisposition to blood cancer, a scientist toolbox. The first one of the series organized by Department of Zoology, Maharajas College, Ernakulam. I am really honored to introduce Dr. Parvati Venugopal, the research person for the day. Postdoctoral Fellow, Department of Genetics and Molecular Pathology, Center for Cancer Biology, University of Adelaide, South Australia. Dr. Venugopal has come to the field of research never by mistake. Research is in her blood and to her bones. With a strong passion for research, she took her master's in regenerative medicine, and from the starts her journey into the amazing world of cancer research. She was awarded Adelaide Scholarship International for her doctoral research, and she was conferred PhD in genetics from University of Adelaide. Dr. Parvati Venugopal is now enjoying the prestigious Maddie Revolts Vision Fellowship to pursue research into the cause and treatment of bone marrow fibrosis. She is the co-inventor on Indian patent application titled Method of Preparing Mesenchymal Stromal Cells for Specific Indication and Composition Thereof. She has published a number of research papers in reputed international journals like Leukemia, Hematologica, Journal of Pediatric Child Health, Cytotherapy, and so on. She is a member of various international societies. The list of her academic accolades does not end. On behalf of Maharajas College, Ernakulam, I cordially welcome Dr. Parvati Venugopal to present the topic. Thank you, everybody. Welcome, Dr. Parvati Venugopal.
Thank you. Thank you for your kind words, ma'am. Also, I would like to thank the organizers for the invitation to present my work here today. Now I'm about to turn off my video because I think it might interfere with the presentation. I will turn on the presenter more. Just a second.
Hello. Hello.
Hello.
Hello.
Hello.
All of you, please switch off your camera and microphone and don't press the present now button. Hello.
Just a second. I'm trying to share my screen.
Okay.
Just a second. Please bear with me a second. I'm just trying to share my screen. Are you able to see my screen now? Hello?
We cannot see your screen.
Just a second, sorry.
Hello? Hello?
Hello?
Hello, biotech presentation. My presentation is on.
Okay, can you see this now? Can you see my screen now?
Yes, it's okay now.
Okay, so everybody is able to see my screen and hear me clearly?
Yeah.
Yes, okay. All right. So today I'm about to speak about my work, and along the way, I will be touching upon some of the tools and techniques that we use to get to some of the results and outcomes that we have come to. Now, just as a start, just to sort of give some basic introduction, as I don't know, I think I'm talking to a pretty diverse audience. So just to introduce quickly, I've got a few introduction slides. So we all probably know that, our DNA or all of the entire instruction booklet to make an organism is encoded within the DNA. And this DNA is packaged into chromosomes, which is found in the nucleus of pretty much every cell in our body. Now, DNA is a long string of 4 biological letters or nucleotides, A, G, T, and C. And a gene is a sentence that is like a recipe to make a protein, right? And the ingredients of this protein are encoded in 3-letter words or codons. Now, within the human genome, we have 3.2 billion nucleotides. And within these nucleotides, we all have genetic variation. By genetic variation, it means like this example that I've shown here in this cartoon. Each of us have variation, we have variance. So for instance, in this example, this person has an A at this position, while another person might have a G, and a third person might have a T. So in each individual, there's about 4 to 5 million genetic variants present, and these are single nucleotide polymorphism, essentially a difference in a single nucleotide. Now, this here is the structure of a gene, and these dark blue boxes show you the coding regions or the exons of the gene, and these are the intronic regions. Now, these SNPs that I talked to you about, these single nucleotide polymorphisms, can be present either within the coding region or the non-coding regions of the genome. Now, majority of SNPs do not impact our health or development, but when these SNPs occur within the coding regions of the gene, it has a potential to impact the protein output from this gene. Now, I've put forward a very simple example of how this might work, right? So what is the difference between a single nucleotide polymorphism and a mutation? Now, this here is the example of a sentence. I've just used a simple sentence to illustrate. Now, when it is a biological sentence, like I said, it's 3-letter words, so it's a frame of 3 letters, how the sentence is read. Now, if you have an error like this or like this, it can lead to it's a spelling mistake, and it actually changes the meaning of the sentence either a little bit or a lot. And this is how a missense mutation is, right? Now, if you delete a single nucleotide, now that changes the frame and the remaining of the sentence becomes meaningless. This is an example of a frame shift mutation. Now, if you put in a full stop early on, then you lose the rest of the sentence, meaning you have an early stop or a truncated version of the protein. Now, these are all different kinds of mutations. So essentially, DNA variations that affect the function of a protein made from a gene are called mutations or pathogenic variants. Now, in our lab, we have a deep interest in studying families with multiple individuals who have blood cancer. This here is a pedigree of one such family. And what you can see is all these individuals that have been highlighted in these blue circles have different types of blood cancer or leukemia. And we are interested in finding out what the genetic cause of leukemia in such a family is. So my supervisor has started a study known as the Australian Familial Hematological Cancer Study, which was established in 2004. And we recruit such families like the one I showed you previously, into the study. And the aim of our study is to do comprehensive genomic analysis on these families and try and answer the question if there's a familial genetic defect that causes this predisposition to cancer. Now, just to quickly introduce you, these are all the cell types that are seen in our blood, right? Now, blood production actually happens from blood stem cells, which live within the bone marrow, which is found within the long bones and of our body. Now, within the bone marrow, you have these hematopoietic stem cells or blood stem cells, which have the ability to self-renew, which means when they divide, they have the potential to create daughter cells that are identical to them, as well as daughter cells that progress in the differentiation pathway and become short-term hematopoietic cells, and further down to progenitor cells, which finally give rise to all these different blood cell types in the body. And this is an essential mechanism that has evolved in living beings, because during the course of our lifetime, we need to have constant production of blood cells. We produce millions and millions of blood cells. So we need a bank of stem cells which can constantly give rise to all these little cells as you go forth. Now, in leukemia, what happens is you have a mutation that can affect any of these stem or progenitor cell stages. And this leads to an abnormal accumulation of cells and uncontrolled proliferation of these cells, filling up the bone marrow and impacting the production of all these other cells. And that's what happens when you have blood cancer. Now, just to put this simply, so when we are born, let's assume that we all have normal cells, and this here is representative of blood cells. What happens in leukemia is there's an accumulation of mutations within some of these cells, and that suddenly leads to uncontrolled proliferation. So also, I would also like to illustrate the difference between germline and somatic mutations. When a person has a germline mutation, it's a mutation that you inherit from either of your parents, and it's present in every single cell of your body. But during the course of our lifetime, we also acquire mutations in cells sporadically, and these are only found in a subset of cells within our body, and these are somatic mutations. So when a person has a germline mutation, it is essentially found in every single cell of their body. And now in the family that I showed you previously, these individuals have inherited a particular gene mutation that predisposes them to blood cancer. So every single cell in their body, including their blood cells, have this mutation. And this all of a sudden predisposes them and makes them more likely to accumulate somatic mutations that are deleterious and can lead to leukemia. So coming back to this family had multiple individuals who had really early onset blood cancer. As you can see, they were all really young. They were in their teens and early 20s. This is really unusual for leukemia because in sporadic disease, people actually develop disease in their 60s. So 65 is the median age of presentation for myeloid leukemia. So the early onset in this family is actually very striking. And this individual is the only person, the only surviving member in this family because the disease was also extremely aggressive in this family. And this person survived only because he had a transplantation, a bone marrow transplantation, which is currently the only completely curative therapy available. Now, at our lab, we performed gene sequencing of 50 candidate genes. So we looked at the coding regions of 50 candidate genes and sequenced them to see if there were any mutations. And we found that there was a mutation in a gene called GATA2 at the 354th position. Now, ever since we published this first family, several other groups have come out and they went and tested their own cohorts of patients, and there has been an expansion of the clinical phenotypes associated with mutations in this gene. And patients actually present with immunodeficiency where their bodies cannot produce monocytes, dendritic cells, natural killer cells, and B cells. And such immunodeficiency is also present with a whole lot of associated infections which are specific to the lack of these cell types. Apart from that, these patients have a predisposition to leukemia, as I described before. Some patients also present with lymphedema, which is due to the defects in the lymphatic system. And there's lung disease, and there's a whole lot of other phenotypes that people present with. Now, what do we know about GATA2? GATA2 is a transcription factor. A transcription factor is essentially a protein that binds, that recognizes to a specific DNA sequence within the nucleus, and it binds to that region, which is usually in the promoter or enhancer region of a gene, and then causes transcription of that particular gene, meaning that gene will be expressed in response to the binding of this transcription factor. Now, GATA2 is a transcription factor which recognizes the GATA sequence, hence the name GATA. And it is known to be really important in the proliferation and survival of hematopoietic stem cells or blood stem cells, like the ones that I described earlier. And it's also known that the level of GATA2 is really important for blood production. People have made mouse models where they have deleted both copies of GATA2, and they have shown that these mice are not able to survive beyond gestational day 10 in mouse. So they're not even born. They actually die from severe anemia. So what we have done, so as I go along, how the mutant version of the protein differs from the wild type version of the protein in its capacity to cause transcription, right? So we used the CSF1R promoter, which is known to be regulated by GATA2. And downstream of that, we have the luciferase gene. So essentially, if GATA2 binds and changes the expression of this protein, it can be measured by measuring luciferase, right? Now here, what I've highlighted with the red arrow is the wild type protein. So we have essentially taken two pieces of DNA. We've taken the mutant or the wild type protein, as well as the CSF1R promoter with the luciferase gene downstream, and transfected that into a cell line. And then we have performed this assay where we've measured the production of luciferase. So this is the level of luciferase produced in response to the wild type GATA2. And you can see there is a significant reduction in this mutation, which is the mutation of our interest, which I showed you in the family. So here we've used cell transfection and luciferase assays to show that this particular mutation, which essentially is a single nucleotide variant, can have an impact on transcription. But the next thing we did was we also wanted to see, so GATA2 is, like I mentioned, it's a transcription factor. It has two zinc finger domains, which bind DNA. Now we wanted to see if the binding to DNA is impacted by this particular mutation. So what we've done here is we've used a technique known as the Western EMSA or WEMSA, and this measures binding of the protein to DNA, like a DNA oligonucleotide which contains a known GATA binding element. So this here shows how well the wild type protein binds to the oligonucleotide or the DNA fragment that we provided. And what we see here is that there is a reduction in the binding when the protein is mutant. The next technique we use co-immunoprecipitation. Here what we do is we take all the protein, we make a protein lysate from a cell line that has been transfected with either mutant or wild type GATA2, and we take the protein lysate and we pull down all the protein that is bound to GATA2. So you use an antibody against GATA2 to pull down all the protein-protein complexes. And then we look for, so we do a Western blot to be able to detect other proteins that are potential binding partners of GATA2. And here what we see, so these are two other transcription factors that are known to interact with GATA2, and they're also known to be involved with differentiation or blood production, like I mentioned earlier. So this here shows how well wild-type GATA2 binds to these proteins. And what you can see is, surprisingly, here the mutant actually binds to these proteins more strongly. And what we think here is by binding here, the mutant protein, by binding strongly to these other protein partners might be somehow impacting differentiation. And this could be one of the mechanisms through which it blocks differentiation into monocytes and causes all those immunodeficiencies. Now, we have also made, we have generated a mouse model where we have tried to replicate the exact mutation seen in the humans in a mouse model. So we have used this technology known as CRISPR-Cas9 genome editing and knocked in this mutation into a mouse model. And interestingly, what we see is, so I must specify here that in the human beings where we see this mutation, it is an autosomal dominant disease. So the people have only one copy of mutant GATA2 and they have a wild type copy, and that is sufficient to cause overt disease in the humans. It has never been reported if there are homozygous mutations in humans. So it was interesting to see that homozygous mutations in GATA2 actually lead to some viable embryos. But you must notice that it deviates from the expected Mendelian ratios. You actually have fewer homozygotes come through than you would expect. So we suspect that there is still a certain level of embryonic lethality seen in this model. Now, one of the things we hope to look at in this mouse model is we want to harvest stem cells from this mouse. So you harvest the bone marrow and plate the stem cells in specialized media in a cell culture dish. So you provide it with all these growth factors and try to promote differentiation of these stem cells into the various different blood cell types. So this schematic here shows you the different cell types that you can measure. And for GATA2, we are especially interested in macrophages and neutrophils within this. The macrophages, because we know that in humans they have a reduction in the number of macrophages and monocytes. So this is something we would like to measure in this mouse model to help better understand the mechanism of disease. And interestingly, these are very preliminary results, and we're still working on this model at the moment. But interesting thing was we saw an increase in the number of granulocytes in this mouse model, and this is also consistent with what we see in patients. The patients who present with blood disease often show a good maintenance of the granulocyte cell numbers compared to the patients that present with sporadic disease. So this is something we will be characterizing further in our studies. Another thing that we are interested in doing, this we haven't done yet, we will be doing in future, is we know that during the course of our lifetime, we are exposed to all these viral and bacterial infections. And that is especially true in the case of these GATA2 patients, because they are exposed to a whole bunch of infections specific to their immunodeficiency. So we would like to simulate viral and bacterial infections in this mouse that mimic viral or bacterial DNA. And then we will be following this mouse model over a long time to see if this results in either immunodeficiency or it results in blood cancer. Now, why does all of this matter? By understanding the mechanism of disease, how a mutation causes disease, we can try and use it to design better therapeutic interventions, which is a major gap in this field at the moment. The other thing, like I mentioned earlier, is for these patients who have really aggressive disease, the best curative therapy currently available is bone marrow transplantation. Now, I'm sure all of us have heard about bone marrow transplantation somewhere, but just to put it really simply, when a person arrives with disease, what normally happens is if they have really aggressive disease, and they want to have a bone, and their only options to have a bone marrow transplant, they're given medication to wipe out the current, all their stem cells in their bone marrow. So they're given the chemo radiation. And once you wipe out their bone marrow, you transplant it with normal bone marrow from a donor who is a perfect HLA match. And this is a highly risky procedure because the procedure of bone marrow transplantation alone has a really high mortality rate. Because there are all these complications associated with the procedure from which the patient can die. And moreover, finding an HLA match is actually really, really difficult. And your highest likelihood of finding a match is usually within your own family. And that is where these individuals with germline mutations have to be especially careful, because it's really likely that all their siblings, or at least some of their siblings, may have the mutation. So that's where it's important to identify the gene and the mutation causing this predisposition within the family, because if you know what is causing the disease, you can test all the potential donors from within the family, and you can select a donor who does not carry the mutation and is also an HLA match for bone marrow transplantation. And this is potentially curative to this patient. Now, moving on to another story. So that was the GATA2 story. Now I would like to switch gears and talk about another disorder known as Diamond-Blackfan anemia. Now I'm going to be talking about a young boy who is 11 years old now. He was diagnosed with this disease when he was 5 weeks old, when he had extremely severe anemia. And when he was diagnosed, all they knew was he has Diamond-Blackfan anemia, which basically means that his body did not have the capacity to make red cells. So in order to live, he had to have regular red blood cell transfusions for the first 3.5 years of his life. And interestingly, when he was about just under 4 years old, he went into this phase where he spontaneously recovered from his disease. So all of a sudden, he went from needing regular red cell transfusions to being transfusion independent, meaning he did not need donor cells to be able to survive. His body somehow started to make its own red cells. And this was a really striking phenomenon. At this point, the family, this here is the young boy with Diamond-Blackfan anemia. None of his other family members had disease, and they all are normal. And this family was really puzzled by what the genetic cause of this disease was. And they had gone through a really painful process of trying to find what the genetic cause was. They had done all these different, like on the family, all these different things were performed on samples from this young boy, where they did single nucleotide polymorphism arrays, whole exome sequencing. Whole exome sequencing is where you sequence the entire coding, all the coding regions of the genome, as well as whole genome sequencing, where the entire genome was sequenced, looking for mutations in genes that are known to cause Diamond-Blackfan anemia. Now, what I must mention here is Diamond-Blackfan anemia is known to be caused due to mutations in the ribosomal proteins. And by looking for mutations using these techniques, they were unable to come to a causative solution. At this point is when this family actually was enrolled into our study, and we performed a high-density SNP array on samples from this young boy. And I would just like to show you how to read the data that comes out of this high-density SNP array. Now, this here is a copy number chart. Now, if we all inherit one copy of a gene from our mother and one copy from the father. So essentially, we should have 2 copies of genes. Now here, a SNP array, what a SNP array does is it looks at SNPs across the genome. And you would expect in a normal genome that all the SNPs sit in this middle line, which essentially means that we have 2 copies of the gene. And the SNP array also gives us another layer. So like I said, we inherit 2. There are 2 possibilities of an allele. You can either have, actually 3 possibilities. You can either have an AA, which would sit up here, meaning you inherit the A allele from both parents, or you can have a BB, meaning you've inherited a BB from the parents, or you can have an A allele from one parent and a B allele from another parent. So it's in the middle here, right? Because you would expect a 50% representation. Yeah. So when we did a scenario on this young boy from a sample from when he was roughly 7 years old, we found that he has a deletion in a small little region on chromosome 12, which sits over here, and this encompasses 11 genes. And interestingly, this includes RPS26, which is a previously reported GBA gene. So essentially, what has happened is within his genome, there is a deletion of 184 kilobase region, which includes RPS26. So he has only one functional copy of this gene. So the loss of the other copy is what has led to disease in this young boy. But there was also another finding here. Now, again, I would like to remind you the difference between germline and somatic, like I said earlier. The other finding was from the B allele frequency chart. And so this is what a normal B allele frequency chart would look like. But in this boy, in this sample from peripheral blood, what you see, this is again chromosome 12, so the same chromosome where he had the deletion sits around this position. But what you see is instead of having 50% of both alleles, there's a deviation. Now, just to put this simply, what has happened here is he has overrepresentation of the paternal allele, so the allele that he, or this part of the chromosome that he inherited from his father. So what has happened within his body has actually somehow undergone this process where a subset of the cells have corrected the genetic defect that he inherited from one of the parents. And presumably this event would have happened in one of the blood stem cells at an early stage. And because this is a correction of a defect, these cells have somehow got a competitive advantage over the remaining cells in his body which have this deletion. So his body is now trying to correct a mutation that was in his germline. And this was a fascinating finding. And it's, I must also mention that in Diamond-Blackfan anemia, about 20% of patients go into spontaneous remission, which is what happened in this boy. Like I mentioned earlier, he phase where he did not require red cell transfusions and his body started to produce red cells. I think there's one more question from Deepak. Okay, so the question is can I give some information on genetic predisposition to AML M2? Now AML, so the classification of AML basically classifies it. So there's a classification system called the FAB subtypes, and M2 is one of the FAB subtypes. Now, as far as I can tell, at least in the families we've seen, I don't know that a mutation is specifically been linked to or exclusively been linked to one subtype of AML. But I think this, we need to be mindful of the number of patients we're dealing with here. So it's a really small number. Like familial cancer is estimated to be, to constitute about 10% of all cancer. So among all sporadic cancer, about 10 to 15% is suspected to or estimated to have a germline origin. But the problem is this is really underappreciated. Like until recently, it was not, like people wouldn't particularly look for genetic causes. So I think this is something that needs to be looked into a bit closely to see if a particular mutation specifically predisposes you to a particular subtype. The gene that I mentioned earlier, the DDX41 gene, has been associated with erythroid leukemias, but again, this needs to be characterized further. Now, Jovita has asked me what is a hematological malignancy. It's essentially, so hematology, like it's a blood cancer, blood malignancy. So a few people have asked for a contact ID, and I'll leave that with Rama ma'am, and she can pass it on to you.
Any more questions?
If there are no more questions, I would like to thank you all for your attention. Thank you.
Thank you, ma'am.
Thank you.
So don't go, ma'am. Okay. So we will have a formal vote of thanks. Respected Dr. Parvati and all the participants, I take this opportunity to express my gratitude to each and everyone. Dr. Parvati, in spite of her busy schedule, spent time to talk for the first session of this international webinar series. The talk was very inspirative, informative, simple and useful. And from the response itself, it sees that everyone is enlightened by your talk. Thank you so much, ma'am.
Thank you. Thank you for this opportunity.
I would like to extend our sincere thanks to Dr. Jayamol KV, Principal, Maharaj College, Ernakulam, for her inaugural address. I express my sincere gratitude to Dr. Emma, our HOD, for giving me such an opportunity and valuable support for the smooth conduction of this webinar. My sincere thanks to Tojo for his valuable support, advice, and also finding time for YouTube live streaming. I express my sincere gratitude to my student, Balindu, for spending her time for making the poster and all the necessary things. I express my sincere gratitude to all my colleagues of the department and other departments of our college for their valuable support and timely advice. My deep sense of appreciation and thanks to all the participants who choose to be live with us and attended the webinar with great enthusiasm and made it a successful event. So thank you all. Thank you so much. And the feedback form is given in the chat box. All of you please fill it and you will get the certificate automatically. Thank you one and all. Thank you.